hear about the project that Tatiana Guerra set up this year at Archbold Buck Island Ranch. So Tatiana has been working at the ranch since last October. She joined in from Miami-Dade College, where she majored in biotechnology. But that was not the first time at the ranch, because her relationship with Buck Island Ranch started earlier than that, because during her bachelor's degree, she worked with Dr. Tamila Burge in a microbiome lab, and she came to the ranch to collect some sample and analyze it in the lab. And you might have seen her before, because she came to our environmental stewardship day to present in the form of a poster. And at that point, she was presenting with Alec, which is over there. So today she's going to talk about plant and microbe association, work she did in our rainout experiment. Later, she's planning to go to grad school, and she's interested in agriculture and biological engineering. So that's what she would like to continue on. But I think first she will just take her time to relax a little bit at home.
Hello, my name is Tatiana. As Greg mentioned, I am going to talk about AMF responses to drought stress through rainout treatment. So some of you may or may have not heard about AMF, or you guys have probably read briefly about AMF, which is good because AMF are ancient fungi that have been associated with plants since plants first colonized land. AMF plays a crucial role in the ecosystem by exchanging inorganic materials such as phosphorus and carbon compounds through mutualistic relationships with more than 80% terrestrial plant species, ranging from bryophytes to tracheophytes. And fun fact about them is that they are obligate symbiotes, meaning that they cannot live without plants because they have lost the ability to synthesize lipids and require precursor molecules from plants. Now that I mentioned that they cannot live without plants, even though they can't, fungi still have some importance and beneficial structures to the plant. So the host plants provide a carbon compounds to AMF in exchange for inorganic minerals and protection against environmental stresses. They play an important role in phosphorus absorption, circulation of nitrogen, in ecosystem tolerance to heavy metals and saline conditions, and increasing protections from nematodes and root diseases in the rhizosphere. They also consider a natural biofertilizers, which are eco-friendly and contain these microorganisms that naturally enrich soil fertility and boost crop growth and plant growth. They offer a sustainable and cost-effective alternative, and this is important to know because about AMF, because since water scarcity is becoming a major global a major global environment problem, it is predictive since there is an increase on global warming, it could lead to severe drought. And that's what I'm talking about this topic about drought stress. And there's a drought limit to the growth of plant and further impact in plant functions, community composition. And to further analyze about their structure, the AMF structure is that plant roots releases a signaling molecules that stimulate fungal spore germination and branching. In response, the AMF produces its own signals that the plant recognizes, triggering changes in plant root cells, signaling cascades in the plant, involving a calcium signaling and genetic activation and facilitates that symbiotic relationship that they have between plant and fungi relationship. After signaling, the fungus colonizes the plant root cortical cells, a structure called arbuscule, which is the figure that you see in this picture of the AMF arbuscule. Further into this presentation, I'm going to show a microscope slide, microscope slide and a picture so you can see how it looks like. I think it's pretty interesting to see how they look. And it does look like a tree branch. And under severe stress, it boosts the plant's resistance by using antioxidant enzyme genes to encounter this stress damage and increasing these peroxidase activities and reduces cytokinin concentration to protect what they already have. So upon reading about AMF, I had these two research questions that I was wondering, and that is, how does drought intensity affect AMF root colonization in dominant pasture grass? And does above ground biomass influence AMF root colonization in the dominant pasture grasses that exposed to drought stress? And I had these two hypotheses. AMF root colonization would increase under moderate drought conditions due to enhanced plant dependence on mycorrhizal symbiosis. And for my second hypothesis is that AMF root will have a significant impact on the above ground biomass of the grass species under drought stress. For my methodology, I would speak about the root collection, root staining, and microscope viewing. For my root collection, I have targeted Paspalum notatum, or as you guys may know as Bahia grass. And it's because it is the most dominant grass species in Buck Island Ranch. In Buck Island Ranch, I had collected and improved pastures in some of the rainout plots, such as pasture F, J, E, and I. I've only collected in high grazes areas and some of the rainout treatments, such as the RQ, IQ, and control. And I've collected during the time frame of January through February, and note that in these months, it was a dry season. If you don't know what is RQ and IQ, RQ is reduced quantity, that it is a shelter or a roof that reduces water input those grass. And IQ is the opposite of that. It receives an additional water. So you can see that this is the roof, and there is water that intakes into the pump, and there are some sprinklers there that comes out and shoots out water, so there can be an additional water. And then there's a control that has obviously that maintains the ambient condition. For my rainout collection, what I did is I go to these pastures and I put in a PVC onto the ground. I picked up a hand trowel and I dig it up, and I have it in this circular shape, and dug it all the way up to 6 inches and picked it up with the soil and root, where I can obviously see that I have fine roots available, so I can further process in the ranch lab. Once I go to the ranch lab, I gently wash the roots to remove the soil, and I separate them and select the fine roots that I see. And once I have those fine roots, I put them in the histocassette labeled in an alcohol-resistant marker. And then I clear the roots by boiling them into 10% potassium hydroxide for 30 minutes, and then acidify them in 3% hydrochloric acid for 2 minutes. And then I will stain them in trypan blue and lactoglycerol. For my microscope viewing, I had mounted 4 segments fruits into a glass microscope slide. And I had put 2 drops of lactoglycerol, and I would go ahead and look at them under the microscope, and I would see that there is a dark blue spores body, as you can see in figure A, with a hyphae extending into the root, as you can see here, and forming in arbuscules. And if you can see the arbuscules, it's like a tree branch from the first or second slide, which is very interesting to see that it's very visible that this is an arbuscule that you're seeing. And then obviously the vesicles. With this, I have counted the spores, arbuscules, hyphae, and vesicles with a clicker in the lab. And if you're curious, I've used 40x magnification, and each of the roots are about 62 millimeters. For my results, I have used one-way ANOVA to see the effects of from the rainfall treatments on soil moisture, AMF structures, and above ground biomass. And it's very interesting. First, I examined whether the rainfall treatment affected the AMF abundance. Although the reduced quantity, or Q, treatment appeared to have a slightly higher arbuscule count than the increased quantity and control treatment, there was a substantial overlap among treatments. And there's also this statistical analysis that showed no significant differences, suggesting that there is a altering rainfall quantity that did not directly influence AMF colonization. And this is from analyzing the hyphae, vesicle, and the spores. And I've also analyzed the other AMF structure, and there was no significance at all. From this, I had then further deep went and checked the arbuscule counts differ, if there was an arbuscule count differentiating amongst the pastures. There's no statistical differences, but as you can see, pasture I is very, very low in arbuscule count. And you will see later in the slide, maybe there is a reason why there is a low count of arbuscule, and there is like a high count of arbuscule in pasture E. But yet again, statistical analysis, there is no statistical significance between them. So this is the soil moisture that varies among pasture sites between December 2025 and February 2026. Pasture I and J has a high soil moisture levels than E and F. And as you can see, pasture I has a very, not very high soil moisture, but this could probably indicate why I in this box plot is very low because of the soil moisture that is going on. And then in pasture E, there is kind of like a dryness in the soil. Thus why there is a high count of arbuscules in there. And then I further went and evaluate the relationship between the AMF colonization and plant growth. I analyzed the Bahia grass dry mass as a function of AMF abundance, sorry. And the blue regression line may seem like a slight negative slope. However, there is no statistical significance between the relationship of dry mass and AMF. In conclusion, in my hypotheses on effects on drought stress, there is some kind of support, but I will further explain why there is that there isn't that much statistical power for it to have this support. And obviously from the previous graph, this is not true. AMF, from the AMF root colonization of the impact of above ground biomass under drought stress. For my future directions, this may explain why there is no significance in my box plot and my linear model. Is because I did not have a large amount of samples, which it would be an ideal thing to do, to have a wider variation to minimize sampling error and increase statistical power. And also it's important to test out the organic matter, nitrogen and potassium, to dictate how AMF plays a role in these nutrients when there is a drought stress. And if you have noticed, I've only collected in high grades and in improved pastures due to time. So it will be great to have a comparison with high grades and low grades, and have a comparison with improved pasture and semi-native pasture to further observe the differences between them. And this is my acknowledgement on everyone at the ranch and even in plant ecology lab for letting me use the chem lab even before the grand opening, and other funding sources. Thank you. Any questions?
Nice presentation. Yes, you were the first to get to use the new lab, I think. I was curious, what is the bahia grass, or what was the bahia grass doing at that point? Was it in a period of growing, like were these new roots, or was it like the end of the season growth?
No, bahia grass at the ranch is like the most dominant species, so it's been there since.
But at the time you were like the month that you were sampling, was it actively growing, or was it starting to die?
Oh, it was growing, but during like the first time of February, it was dry. So there is some kind of, might be a reason why there is no significance in above ground
So
biomass.
drying out, they're probably older roots, which would have had been more colonized versus if they're like brand new roots, then sometimes you see less colonization. We had enough time. So
Yeah.
this is good context for what you're seeing.
Yes.
I guess I understand the challenges with the short time and trying to get enough replication. So I guess I'm a little unclear in the study, what kind of more replication would be helpful, you think? Would it be better to have like more plants per plot or more plots? I'm just not sure. I guess maybe I missed like sort of the basic design of how many, like there were roots, plants, and plots, but I'm not sure what the actual replication was.
I only did one replicate, which is why, like I said, there is.
Okay.
Yeah. But the ideal is to have multiple replicates of the root samples, especially like in, I would say every 2 months.
Oh, okay.
Yeah. I think that would be an ideal root collection
Right.
time.
Right. There's just like there'd be variability probably at every one of those levels between plants.
Yeah.
So
I think
it's
it would,
challenging.
yeah, and I think it would be great not to collect dry season, but also wet season to see the difference between them, and how much root colonization, AMF root colonization there.
We have a question online. How much rain, so how much rain was happening during the study?
Oh, it was dry season. There was little, no rain.
Yeah, when we say dry here, we mean dry.
Dry.